I don't know. So I ended up drawing everything this morning. And we're ready to start the cerebral lobes, cerebral hemisphere. So we're on page 60 of your lecture notes. And hopefully we'll have it back by tomorrow. There's a cord that's missing that somebody ran off with. So it's not something that I can put the right things back together. Whoever was teaching last night or yesterday will be able to fix it tonight, hopefully. So if you recall, just do a little bit of review. We started out with the neural tube. And then in our short embryology discussion, the neural tube expanded in the cephalic or head region into 3 vesicles. And the first of those 3 vesicles is called what? Excellent, prosencephalon. And the second vesicle, mesencephalon. And the third vesicle, rhomencephalon. Mesencephalon would be the second set of development. So today we're going to look at the development of the prosencephalon, all right? And then tomorrow we'll pick up mesencephalon, rhomencephalon tomorrow and Monday. We're a little bit ahead of schedule. So the prosencephalon developed these elephant ears, if you recall, and the space within the elephant ears is what ventricular system? Lateral ventricles, okay? So these 2 subunits are the telencephalon and the ventricle associated with the telencephalon is lateral ventricle. And the telencephalon becomes our cerebral hemisphere. So we're going to look at the occipital lobe, parietal lobe, temporal lobe, frontal lobe, and insula. And deep inside the lobes, we're also going to have the cerebral nuclei. So that's telencephalon. Okay, and that's what we'll talk about before break. And then this region is diencephalon. Good. So the prosencephalon further develops into 2 structures, and the ventricle of the diencephalon is, well, that's the structure. Oh, okay. So we'll go ahead and put that here. Third ventricle. And thalamus and hypothalamus. And without the PowerPoint, I kind of left the discussion a little early anyway. Tomorrow we're talking about diencephalon. So today we won't cover thalamus, but we'll talk about hypothalamus. We'll talk about thalamus. And in addition, we have the pineal gland, which grows out of the roof of the hypothalamus. Okay? So that's kind of what we're going to focus on today and most of tomorrow. And then we'll start and finish tomorrow with Tuesday, with brainstem. And that's our mesencephalon and rhomencephalon structures, our cerebral aqueduct and 4th ventricle, okay, with those components. All right. So let's do the brain now. Draw the So what is this sulcus right here called? And you're welcome to get brains again from the shelf if you wish. Lateral sulcus. The groove between the frontal and parietal lobe is lateral sulcus. And then about just past halfway, we have the central sulcus. And then we can see the beginnings of, but the rest of it is on the medial surface. Separating parietal and occipital lobe, we have the parieto-occipital sulcus. Right, which brings us to the first lobe that we're going to describe, and that is the occipital lobe. Right? So as we go through each of these lobes of the cerebral hemisphere, you have boxes that have component parts and describe their functions. And so for lecture concepts, you need to recognize what structures are found in what lobes. So if I say the primary visual cortex is a component of which of the following regions of the brain or brainstem, or all of the following are components of the frontal lobe except. So similar type questions to the way we talked about bones and their processes and foramina. Okay? So when we look at the brain, these functions were found sometimes through problems, diseases, and tumors and lesions that people lost a function because of a blocked blood vessel, a stroke. But more detail was found during brain surgery when patients were awake and had their skin and skull anesthetized but were conscious. And so when various regions of the brain surface were stimulated with an electrode, they responded by saying what they felt or what muscle region moved. Okay? So let's use blue for the region of the occipital lobe. So this area is the occipital lobe. And you'll recognize the names of the bone components of the cranium. I'm not sure which came first, the chicken or the egg here. The occipital bone and the occipital lobe are both together. And there is an area kind of here at the tip that is known as the primary visual cortex. So the gray matter and these gyri, or components of the gyri, are formed of cell bodies which are churning the action potential, the electrical current coming from or originating in the retina in your eye into an image. All right, so you have various rods and cones picking up various shades of color in light and dark that are being activated by photons. So when I look at this tabletop, all right, a series of waves of energy are entering my eye, activating at various wavelengths the cones in my eye, creating signals that are just electrical current sent back to my occipital lobe, where different regions are going to be activated and process that as a depth, a light, a shape. Now I can do that with my eyes closed. Ever done that? Made little movies on the back of your eyelids? Now I wear contacts or soft contacts so it doesn't sound too uncomfortable. But I can sit here and push on my eyeball and make little light shows, like a laser image on a clip. Maybe not as fancy. But what I'm doing is putting pressure on the retina, and that pressure is causing the same change in the opening or closing of channels and triggering a current. And my brain doesn't know the difference between a current coming from pressure on my eyeball versus energy in a photon. Okay? An electrical stimulus, an electrode on that primary visual cortex, will cause a light burst. That's why you see stars when the occipital lobe is compressed. And I remember reading about this individual, this guy who was blind, and he knew Braille. So with his fingertips, he could detect a pattern of raised dots and interpret that as letters. And he had surgery done and had electrodes implanted in his primary visual cortex. And there are special computers that will read books. You just lay the book on top, and it will read in this computerized voice. And instead of reading out loud, it sent electrical signals through the electrodes into his brain, and he saw light patterns as Braille patterns. So he didn't see the words, he saw dots in a Braille pattern that he visually read rather than using his fingertips to read. Now, I don't know that that was more than just an electrical experiment. I mean, what the advantage was to having that occur. I don't think he was deaf, but it was just showing how the electrodes can emulate that area. All right, then we have an association area, which is going to help you make sense of what you visualize. In other words, the primary visual area is going to just create the image. All right? So, for instance, a photograph camera takes a picture. You have a digital camera, and it takes a picture. It processes the energy coming into the camera to take that picture. But the camera, that picture doesn't mean anything to the camera. All right? It's just, it creates the pattern of reds and blues and yellows and whites. It means absolutely nothing to the camera itself. And that's what the primary visual cortex is doing. The association area is where you compare that image to other images, as well as use long association fibers with your frontal lobe to recognize the meaning of that image. All right? So when I see the tabletop here, I recognize it as a wood pattern. I know it's a tabletop. I could name it using association neurons with my frontal lobe and words. That requires the visual association, okay? To make sense, to associate the image with some other region. All right, then we have the temporal lobe, and the temporal lobe is separated from the frontal lobe by the lateral sulcus. In addition, if you look on the inferior aspect, if I can borrow the brain part. So here would be the occipital lobe. Here's the parietal occipital sulcus. All right, and this part that I'm grasping with my fingers, both medial surface and lateral surface, is occipital lobe. That's going to be supplied by the blood of the posterior cerebral artery, and pica, posterior inferior cerebellar artery, will do the lower portion of it. Then if we turn it laterally, here's the lateral sulcus, and this is temporal lobe. Right? Temporal lobe is also this inferior region. And so we don't have temporal lobe in our medial surface, but we do have it down in this region here. Okay? So, the inferior aspect. Temporal lobe is, the sensory aspect of the temporal lobe is primary auditory, and on its deeper surface, taste. But this would be the primary auditory area. So remember the internal acoustic meatus? That's where the nerve from the inner ear comes into the skull, and it's going to send its information through the thalamus to the primary auditory area where interpretation of sound occurs. So whether it's a bell ringing or someone knocking on the wood table or footsteps, that processing, changing the electrical current to sound, occurs in the primary auditory cortex. And then it also has an association area. Which requires memory being laid down. Okay? Have you ever heard the first notes of a song and then your brain fills in the rest? All right? You don't hear that like you would hear it if it was actually being played, by an orchestra or a band. but your brain fills in those notes. So I was taking a music appreciation course, a humanities course when I was in college, and this was back when we used records, not CDs even. And every time the professor started to play a particular song, it was classical music and it was by Mozart, when he put the needle on the record, it would skip every single time. And then when he played it for our exam, the same thing happened. The needle skipped. And before the music even started, because of that sequence of repetition that had occurred, my brain filled in all the rest of the notes of that song. And I knew what song it was going to be before he even started playing it. All right? So those earworms, you guys know what earworms are? It's when you hear a catchy tune or an advertising tune or whatever, and you can't get rid of it. It just keeps playing over and over in your mind. So that's using the auditory association area. All right? Certain songs have memories. So at weddings, they'll play the my song, or the couple's song, and then after that, if it's a good marriage, it'll bring back happy memories whenever they hear that song. Okay, so that involves the auditory association area. Now, in the cortex of the temporal lobe, and since I don't have the image projector, let me draw a coronal section. So this is the longitudinal fissure, and we're kind of in the parietal section here. So this would be the lateral sulcus right here. And then down here we would have the temporal lobe. And then, so this would be temporal lobe. And I'll just go ahead and put in the lateral ventricle. And the third ventricle here. So in this region of the temporal lobe, the gray matter is kind of coiled in a tight coil like this. And it's in the floor of the lateral ventricle, the inferior horn. Let me show you. Let's not fill this in completely. Kind of hint at the CSF fluid in here. All right, so what I'm talking about is, remember this represents the fluid in the ventricular spaces? So we've sectioned through the brain right about this level. So this portion of the lateral ventricle that I've drawn there, and then this would be the inferior horn. So we're seeing the lateral ventricle here and down here as well. Okay? So the lateral ventricle is curving out and back in. So that's why we're seeing it in two places. Well, or front, just start together. All right? So this area of coiled gray matter is the hippocampus. And it's found within the temporal lobe. And it's involved in the formation of short-term memories. There There was a sailor back in World War II, kind of at the end of World War II, who liked to make wooden ship models. And we didn't have the fast drying glue, so he had to sit there and hold these delicate parts of the ship for a long time, and his roommate liked to tease him because he knew that he couldn't do anything about it because he was holding on to these glued pieces of wood, and he would poke them and otherwise, try to frustrate him. And so one afternoon, the guy got really mad, and he just whirled around on his roommate who had an ice pick that he was jabbing him with, and it went right through his skull, into his temporal lobe, through the hippocampus. And he lost his ability to make those memories any longer than 3 or 4 minutes. So for him, it was the frozen in time. The president was always the president. Who was president at the end of World War II? Truman, in the late 40s, okay? And you walk into the room and say hello and shake hands and, oh, how are you? And then 3 minutes later, it was as if he'd never met you before. And so when he died, his medical doctors had gotten him to agree to donate his body for autopsy, and the hippocampus had shrunken. There's also changes in the hippocampus seen in Alzheimer's or in certain forms of senile dementia. Interesting, for him, it was just the one side. So there's some one-sidedness, just like there is to our speech centers. Hippocampus refers to seahorse. Hippo is horse. And that's the coiled nature of the gray matter that's found there. So when I get my PowerPoint projector back tomorrow, we'll spend some more time looking at this. And then on Tuesdays, when we need our modeling clay, and we'll be modeling these structures before we find them in the sheet grain on Thursday. The last structure to talk about here is associated with both the parietal lobe and with the temporal lobe. So I'll draw its colors with both. And this is, the old name for this is Wernicke's area. And now it's more commonly known as sensory speech area. And this is the area associated with the interpretation of speech, right? So if I say cat, what happened in your brain? You heard the word cat, right? But you also now know what I'm talking about when I say cat. And if you have a cat, you probably saw your own cat. If you ever got scratched by a cat, you might think of the noises that a cat makes, whatever. Baby kittens, okay? However, if you had sensory speech area damage from a stroke or a tumor of some sort affecting that area, then you would be able to hear the word cat, but it wouldn't make any sense to you. All right? Let's say that the word mork meant cat. And I said, "Am I mork the other day?" You wouldn't believe, and you heard mork, and you wouldn't know what in the world I was talking about, okay? Because you wouldn't be able to interpret it. And that's what happens when the sensory language area is damaged. Sometimes it's just a few words, sometimes it's the entire language. They can hear it, but it doesn't make any sense. And it's all forms of communication. So writing the word cat doesn't make it any clearer than the sound of the word cat, or sign language. Now if you showed them a picture of a cat, all right, they would see the picture of the cat, but that symbolism, where one thing means something, they can't do that interpretation, all right? And so it's, like I said, it's variable. It may just be certain words. It might be, whole concepts, but that makes it difficult for them to understand. What they say doesn't make any sense to them either. All right, they know what they want to say, but they don't know if what they said is what they want to say.